So this lecture will cover different types of neuroglial cells. Glial cells are the helper cells of neurons, and they're present everywhere around neurons. So the types of neuroglial, there are 4 types of cells, neuroglial cells in the central nervous system. And the first type is astrocyte. So these star-shaped cells are called astrocytes. And then you have the ependymal cells. The ependymal cells are like the epithelial cells around the central nervous system, and they secrete and produce cerebrospinal fluid. And the next type of neuroglial cells is the microglial cells. So the microglial cells look for debris and pathogen, and they basically perform the job of phagocytosis. So they engulf any pathogens and destroy them. And the 4th type of neuroglial cell in the central nervous system is the oligodendrocyte. Now the oligodendrocyte form the myelin sheaths. So this sheath that you're seeing on top, so this transparent sheath that you're seeing on top of the neurons are the myelin sheath. And the main role of the oligodendrocyte is to make the insulation that you see around the axons of the neurons. Okay, so these are called oligodendrocytes. So these cells right over here. So oligodendrocytes, you can see, are kind of wrapped around the neurons because they form the insulation around neurons. Now the first type of cell, which is astrocyte, are the star-shaped cells. Now star-shaped cells called astrocytes are most abundant in the glial cell in the nervous system. They cover the brain surface and non-synaptic regions of neuron, meaning where another neuron doesn't meet a neuron. So in the gray matter of the brain. Now the astrocyte perform a whole range of diverse function. They support framework, and they have something called as perivascular feet. So these green structures that look like feet holding blood vessels together on one side, and the other side they are attached to the neuron. These are called perivascular feet. Perivascular feet help in forming the blood-brain barrier. So they filter blood from entering the brain, and it restricts the movement of substances between the blood and interstitial fluid. So it basically protects the central nervous system. And another important function of the astrocyte is to convert glucose to lactate. Now, lactate is the only form of nutrient that the neurons can use. So the astrocyte basically convert the available glucose to lactate, which is then taken up by the neurons. They also secrete growth factor for effective communication, and they regulate the composition of the tissue fluid by absorbing excess neurotransmitters and ions. So they help in conduction of efficient nerve impulse. Now when the astrocytes are damaged, it's called astrocytosis or atherosclerosis. Now astrocytosis form hardened sacs of scar tissue in the filled space, right? So we call it astrocytosis or sclerosis. Sclerosis is basically the formation of scar tissue. All right. And they also play an important function in learning and memory is what has been found. So when researchers saw the brain of Einstein, he had a lot more astrocytes than a normal person. And that's when they realized that the astrocytes are special. So they are big and bushy with so many branches. So they were called astron or the star cell. So this development in 1997 was important in understanding the role of astrocytes. Until 1997, we didn't really know much about the role of astrocytes in the brain. Okay, so the next type of neuroglial cell is the oligodendrocyte. Now, oligodendrocyte are the cells that form myelin sheath around the neuron. So they resemble astrocytes, but they are smaller, but they have fewer processes compared to the astrocyte. So so these are the oligodendrocytes. There are fewer processes, but then these processes basically completely wrap around the axons of the neurons, because these translucent gray structures that you're seeing help in forming the myelin sheath that insulate the axon of the neuron. So they help in efficient signal conduction from the neuron. The ependymal cells, that are the epithelial linings of the brain, they secrete cerebrospinal fluid, and they also help in formation of this blood-brain barrier. And this is mainly cuboidal epithelium with cilia on the surface, but they have a special name known as ependymal cells. But in a sense, there is cuboidal epithelium with cilia on the apical surface. All right. So the next cell is the microglial cells. Microglial cells are the phagocytes of the central nervous system. So these macroglial cells basically just float around and they just eat up any dead, damaged cell or any pathogen that is found in the cerebrospinal fluid. So the cerebrospinal fluid is the fluid present in the brain and the spinal cord, and it is of a lot of clinical significance because for diseases like meningitis, by doing a spinal tap and extracting the cerebrospinal fluid, they can look at the cultures or bacteria present in it and diagnose the conditions like meningitis. So outside of the central nervous system, meaning outside of the brain and the spinal cord, you can find two types of neuroglial cells. That is in the peripheral nervous system. The first cell is the Schwann cell. The second cell is the satellite cell. Now these two cells are not found in the brain or the spinal cord. They are only found in the peripheral nervous system. Now the first type of cells, Schwann cells, from myelin sheaths just like the oligodendrocytes in the central nervous system. But these particular cells form myelin sheath only in the peripheral nervous system, which is outside the central nervous system. So they help the damaged cells to recover, and that's why they are the workhorse of the cell, basically. They function exactly like oligodendrocytes would in the central nervous system. And the next type of cell is satellite cell. Satellite cells surround the neurosoma. So these over here are satellite cells. And these marshmallow-like cells that you see wrapped around the nerve fiber are Schwann cells. Now the satellite cells surround the cell body in the peripheral nervous system. They provide a thick layer of electrical insulation So they help in regulating the environment of neuron. Okay. So the glial cells play a very important role in tumors. So mass of rapidly dividing cells that really, once mature, they don't have much capacity for mitosis and seldom form tumors. So neurons itself don't do a lot of cell divisions. Once they mature, that is it. So brain tumors arise from meninges, the cells of the meninges, which is the protective layer, a big, thick protective layer around the central nervous system. So it's a three-layer that makes up, that protects the central nerve, a three-layer protection of the central nervous system. And then you have non-neuronal cells that are tumors that can metastasize in different parts of the body and eventually in the brain. And then often glial cells, but not the neurons, they can actively divide throughout life. So gliomas are often very malignant, and they grow rapidly because they constantly divide. Malignant means deadly because they are capable of constantly multiplying rapidly, and they are capable of metastasizing in different parts of the body. So once gliomas become tumorous, the blood-brain barrier decreases the effectiveness of chemotherapy if it's a glioma. So the treatment consists of radiation or surgery depending on the type of tumor. But chemotherapy itself, it uses a lot of toxic chemicals. So once a person undergoes chemotherapy, it really makes the blood-brain barrier weaker, so a person is prone to more infections. So gliomas are really hard to treat. So if you remember in the earlier chapters, we looked at a gamma knife, which we talked about there being one in Vanderbilt. So the gamma knife is one of the most the common methods nowadays for treating a glioma. So neurons are all excitable, meaning they are capable of irritability. So they respond to external changes in stimuli. And then they are conductive. So respond to stimuli by producing electrical signals that are quickly conducted to other cells at distant location. Now, all cells of the neurons are excitable and conductive because they are capable of responding to an external stimuli, and once they sense the change in external stimuli, they can quickly conduct the stimulus to other cells by transferring electrical signals. And then when the electrical signal reaches the end of a nerve fiber, it secretes a neurotransmitter that influences the adjacent cell to receive and transmit the signal. Okay, so nerve cells from nervous tissue which rapidly receive and analyze signal, they produce an appropriate response depending on the type of signal they're receiving. For example, if you get cold, you get goosebumps, right? But if you touch a hot stove, you want to pull your hand away because you know it's hot. So all of these functions are basically performed by the nervous system. The muscle itself is just the effector that listens to the command from the nervous system and performs the final action. But the communication is solely performed by neurons of the body. There are different types of neurons in the body, and they can be classified on the basis of structure, function, and electrical impulse. So on the basis of structure, there are three types of neurons: multipolar, bipolar, and unipolar. Based on how many processes, so these extension, that those are collectively called axon and dendrites. Based on how many processes emerge from a neuron, we can classify them as multipolar, bipolar, and unipolar. Now, the multipolar neuron, you can see from the cell body, there's a whole bunch of processes emerging from the cell body, and this type of neuron is present in the brain. So they are present in a person's head. The bipolar neuron, you can see, only have two processes emerging from the cell body. Eventually they can split into several smaller nerve terminals, but from the cell body itself, we only have two processes emerging. And this type of neuron form the sensory organs, like eye, ears, olfactory neurons, etc. And the last type of neuron is unipolar neuron. So unipolar neuron has dendrites and one axon that are fused together to form a continuous process. Right? So that's the axon and dendrite that fuse to form one continuous process. Now, the dendrites of unipolar neuron function as sensory receptors as well. Okay, but the bipolar neuron are abundant in the eyes, inner ear, and the olfactory areas of the brain. Everywhere else as sensory receptors, you have unipolar neurons. So here you see some examples of the sensory receptors. This one is the corpuscle of touch. So they sense any touch in the environment. And then you have, these are all unipolar neurons, right? So these are sensory receptors. And this one over here is a lamellated corpuscle. So these are pressure receptors. And these ones over here are nociceptors. Nociceptors meaning pain receptors. So if you get pricked by a pin, the nociceptors are the ones that pick up the pain from the environment. And these ones over here are tactile epithelial cells in the epidermis of the skin, which is the outermost layer of the skin. And these are Merkel cells. That's what they're called. So the Merkel cells or tactile cells that sense a light touch in cells.